the mitogen-activated protein kinase pathway, or MAP kinase pathway. Here we have a picture of the MAP kinase pathway. A typical MAP kinase pathway has the following parts. RAS will go on to activate MAP kinase kinase kinase. MAP kinase 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 will go ahead and activate MAP kinase kinase. And MAP kinase kinase will go on to activate MAP kinase. This will lead to the activation of downstream effector proteins by MAP kinase. In humans, the MAP kinase pathway is as follows. RAS activates RAF. RAF activates MEK or MEK. MEK activates ERK1 or ERK2. And ERK1 or 2 will go on to activate downstream effector proteins. RAS activates RAF by binding to RAF. RAF activates MEK by phosphorylating MEK. MEK activates ERK1 or 2 by phosphorylating ERK1 or 2. ERK1 or 2 are kinases that activate downstream effector proteins through phosphorylating them. Among the proteins which are activated by ERK1 and 2 are transcription regulatory proteins, TRPs, also known as transcription factors. These TRPs will activate the transcription of immediate early genes, IEGs. The product of some of these IEGs are also TRPs. Example of IEGs whose products are TRPs are FOS, JUN, and MYC genes. These TRPs, products of expression of IEGs, will activate the transcription of delayed early genes, also known as DEGs. ERK1 or 2 will go on to activate TRPs that already exist in the cell but are in the off position. This is activation through phosphorylation. These TRPs will go on to activate the transcription of IEGs, and within these IEGs exist genes that code for new TRPs. This is activation through transcription and translation. These new TRPs will go on to activate the transcription of the delayed early genes, or DEGs. This is activation through transcription and translation. Now we have this picture from our study guide. You have activation of the RTK. You have activation of ERK1 or 2 downstream of the pathway that's activated by the receptor tyrosine kinase. You have activation of TRPs by this activated serine threonine kinase. You have the transcription of IEGs. You have translation of the IEGs. Included within the products of those IEGs are new transcription factors, or TRPs, and they go on to activate the transcription of delayed early genes, or DEGs. Two IEGs are FOS and JUN. Two products of the FOS and JUN genes are the FOS and JUN proteins. FOS and JUN form a dimer called AP1. So if we look at these two pathways that are activated by RAS, you can see that here is FOS, there is JUN, and that leads to the activation of AP1 through dimerization of FOS and JUN. AP1 is a TRP. One of the genes that AP1 activates is cyclin D. The cyclin D is a delayed early gene. And there's cyclin D1. A simplified but specific pathway can be written for the cyclin D activation. ERK1 or 2 activates, AP1 activates cyclin D. What does cyclin D do? Cell cycle progression and the R point. The cell cycle has the following phases. G1 is cell growth phase. S phase, a phase in which the chromosomes are duplicated. G2 is second cell growth phase. M phase, where mitosis occurs. The restriction point, R point, is at late G1 in the cell cycle. The activation of growth and proliferation pathways such as the MAP kinase pathway will push the cell through early G1. If the growth and proliferation pathways are shut down prior to the cell reaching the R point, the cell will not continue through the cell cycle. If the growth and proliferation pathways stay active, the cell will continue through the cell cycle, and once it is past the R point, it is committed to two outcomes. Completion of the cell cycle and producing two daughter cells, Apoptosis, programmed cell death, if the cell cannot complete the cell cycle and divide. Therefore, cells are only responsive to growth-promoting signals from early G1 to the R point. Once a cell is past the R point, the presence of growth-promoting or growth-inhibiting signals will not affect the outcome. For cells to progress through the cell cycle and through the R point, the concentration and activity of cyclin D must increase. Cyclin D is a DEG whose expression is activated by RAS. Now we can summarize the whole process. Growth factor binds to growth factor receptor in RTK and activates it. The RTK activates GRB2. GRB2 activates SOS. SOS, which is a RAS-specific GEF, will go on to activate RAS. RAS will go on to activate RAF. RAF will go on to activate MEK. MEK will go on to activate ERK1 or 2. ERK1 or 2 will go on to activate AP1. AP1 will go on to activate cyclin D. Cyclin D will go on to push the cell through the cell cycle, and you get cell growth and proliferation.